All right, welcome back. We're going to do concept 4 now. So concept 4 covers eukaryotes. The eukaryotes, the previous video was on prokaryotes. Eukaryotes, I'm just going to give a fairly simple definition, just some simple characteristics. It's a cell which has nucleus and membrane-bound organelles. And then also typically like later cells, they're not as old as bacteria typically. I mean, the oldest bacteria is probably older than the oldest eukaryote. Then again, it all relates back to the oldest cell, that ancestor cell. So yeah, also pretty big cell. Okay, so the three things we're going to talk about in this video are how do we differentiate prokaryote versus eukaryote, then we're going to be talking about how the eukaryote evolved, right? And then finally we're going to be talking about different types of eukaryote. So basically we're just going to give an overview of what is a eukaryote, give an overview of what is a eukaryote, how it differs from prokaryote, how they evolved, because there's a lot to say, and then two different types. There's going to be plant and animal. Okay, cool. So let's talk about prokaryote versus eukaryote. Versus eukaryote. So when we talk about the difference, we talk about prokaryotes being, so I'll say P for prokaryote. P are very simple. E for eukaryote, very complex. If we look at DNA, P have circular DNA. Eukaryotes have one DNA molecule, you know. I gotta set up some better settings for this. Not mole. This happens, pretty annoying. You have one molecule of DNA, basically, for a eukaryote. So eukaryotes have DNA as chromosomes and chromatin. So multiple molecules of DNA. So longer, these ones are shorter. That's pretty important to see. Talk about organelles, that's also very important. Prokaryotes, no organelles. They have ribosomes, but ribosomes don't really count as organelles. Eukaryotes, yes, membrane bound organelles. What else? We talk about plasma membrane. Now, this is a bit of a similarity. Plasma membrane. Like, it's largely the same. Like, they're very similar, but for example, in eukaryotes, they have, and we'll talk about this in another video for more detail, but eukaryotic cells have cholesterol for like rigidity of membrane. Rigidity of membrane, but prokaryotes do not. They don't have cholesterol, but like it's both a phospholipid dilayer, so it's largely the same there. Now we're going to talk about the cytoplasm. So basically cytoplasm, it's fairly similar in both cells. It just depends on like what proteins are used. So cytoplasm is cytosol, so jelly, plus cytoskeleton. So it's like skeleton, that's the best thing. So the cytosol is pretty similar, but cytoskeleton of prokaryotes, cytoskeleton is mostly it's mostly FTSZ. So so FTSZ stands for filamenting temperature dependent. Temperature dependent sensitive mutancy. You can see why it's got an acronym, because it's a terrible name. So cytoskeleton of the prokaryotes is mostly FTSZ. Of eukarya, it's mostly tubulin, right? They have different compositions of cytoskeleton. Let's talk about DNA location. So for P, it's free in nucleoid. And for the eukaryotes, it's in nucleus as chromatin. It's packaged basically. Yeah, the cytoplasms are fairly similar in both cells. Now, let's talk about diversity. That's very important. So the eukaryotes are not very diverse, and the prokaryotes are extremely diverse. They can exist anywhere, but they're very common. They're very simple. So you can think of them as a very simple but very diverse. All right, cool. So that's basically the difference. Chromatin is like DNA molecules, but they're packaged as chromosomes, I mean to say. Okay, so that's the difference. Now we'll talk about just some general structures in a here's a nice little note. Identical structures in P will be more complex. Right? Just the same structures are more complex. Like if you look at flagella, prokaryotes is very simple. Eukaryotes is really complex. Okay, so let's look at eukaryote structures. Okay, so very important. Nucleus. Now, the nucleus itself we could talk about for quite a while, but let's just keep it brief. It's got a double membrane. It's got nuclear pores for flow of stuff. It's got these chromosomes, it's a bit of genetic information. It's got basically, if we look at RNA made in nucleus is transported out for translation. correct term is. Transcribed. Right. Yeah. Now, so the number is unique to different animals. So we have 46. Right, let's talk about the chromosome now. So basically the chromosome is like contains the genetic information. It's one DNA molecule, and it's made up of chromatin. Chromatin, which is a complex of DNA and protein. Also, with the nucleus, there's a part called the nucleolus, which is the site for ribosome synthesis. So ribosomes are made in the nucleolus. That's a terrible lettering, but you get the point. Nucleolus. Okay, eukaryotes also have something called a mitochondria. So it's a respiration, right? It's got a bunch of mitochondria. Oh, one big one. Powerhouse of the cell, to be honest. So respiration happens here. Yeah, so we use glucose to create these structures. I mean, these structures use glucose to carry around aerobic respirations, making energy. Right. So double membrane, very important. So outside, and then we have a inside membrane folded into channels, cristae, and this is maximizing, well, not maximizing, but increasing the what's the word for it? Increasing the surface area to volume ratio, and making the whole thing more efficient. Cool. There are also like chloroplasts, basically. and they do, they contain photosynthetic components. And these photosynthetic components just allow them to make glucose with that very classic reaction, H2O plus CO2, which is glucose plus CO2. Cool. Yeah, so that's what it is. Their structure is a bit, it's exactly, well, you've got a outer plus inner membrane, it's the same, so two membrane. And it's the inner membrane has a network of like folds, folds, like a network of they're called lamellae, or thylakoids. They just exist as stacks, and they have and they contain the photosynthetic pigments, photosynthetic elements. For sunlight. That's the thing. It just ties back, basically. So the thylakoids as well, they exist as stacks. It's like, yeah, they're just like stacks, basically, within the chloroplast. Cool. Yeah. Now, we'll talk now about, so we'll talk about prokaryote, eukaryote. We'll talk about evolution now. So, let's talk about this process called endosymbiosis. So, the thought is that organelles come from endosymbiosis. Sorry. Okay. Basically, what this is cell eats another cell and becomes like becomes part of the other cell. So yeah, I'll just go through the process. It makes more sense. So you have step 1, which is and this is what we believed happened. a long time ago. So there are two types. This is primary and secondary. So primary, eukaryote engulfs prokaryote. Membrane formed. Vacuole is formed. The membrane evaporates and DNA of prokaryote becomes part of nucleus and eukaryote. For you have like new organelle. Cool. So yeah, basically that's sort of the thought. You'll end up with something that has like two membranes around it, because as it came in to the thing, it had two, and then it became a molecule with a cell with three, and then eventually became two again. And that's sort of the process of the that's the process of creating a new organelle, which will be mitochondria in this case. So I just check real quick. So a secondary endosymbiosis follows similarly, except it's the like endosymbiosis. So secondary endosymbiosis when eukaryote swallows I like to think of it as swallows. Engulf is probably a better word, but when a eukaryote engulfs another eukaryote, when that happens, basically you get an organelle with three membranes, right? Because you have there's like a eukaryote, so there's already a double membrane of a organelle in that eukaryote cell. Then there's another membrane, so that's three. And then one membrane forms around when it's engulfed, when the old cell is engulfed. And then that membrane eventually, of the engulfing cell, eventually dissolves. So you're left with either, something dissolves, but it doesn't have to. So you can have three or four membranes. from secondary endosymbiosis. And yeah, so it'll have three membranes at least, thereabouts. It's not entirely clear. Again, it's a pretty good theory, but it's not like perfect. There's also some evidence that use crypto monads, because they have engulfed a cell. So with this primary endosymbiosis, you get a new organelle with two membranes, and with the secondary you get 3 or 4, it depends. But what's important is that gene transfer happens, and that's what allows the eukaryote to become the organelle. So that's what it means for endosymbiosis. Now let's talk about the nucleus. I don't think we really need to go into this too much of this thing. I mean, it's not incredibly difficult. Okay, so nucleus formation. We believe it was, and this is a theory again, lots of theories in science, unbelievably. Invaginations of nuclear membrane. So the nuclear membrane of a prokaryote folded in on itself, and over time that turned into the endoplasmic reticulum, and the nucleus just sort of formed around it, and eventually it separated, so you have that separate structure of a new cell membrane, and then there's those invaginations which create a nucleus. I mean, that's the entire concept. It's not incredibly difficult, I think. Yeah. Now, we need some proof for this as well, for this whole system. So some of the best proof is that the organelles share DNA with prokaryotes, the equivalent prokaryotes. So for mitochondrion, for example, it's a purple bacteria, for example. So that photosynthetic, that mitochondrion that was a purple bacteria, it shares DNA. So that indicates that they're similar in some way. They share a great deal of DNA. So the fact that there is an outer membrane in mitochondrion gives this sort of, I don't know why I drew that like that, a secondary membrane. A second membrane indicates endosymbiosis, because it's from this process, the membrane doesn't evaporate. That's a bit misleading. There's like a thing and it evaporates around, but the main thing is like the DNA of the prokaryote becomes part of the nucleus, and so there's no real separate. The vacuole dissolves, right? The membrane stays, but the vacuole dissolves, as far as I can tell. Indicates endosymbioses. I would also, there's a bit of evidence which says, so they keep machinery, own machinery for biochem. So they keep their own machinery, the organelles. Okay, and they also divide by binary fission. So this is pretty legit stuff, because it's like prokarya. Cool, isn't that cool? We're drawing some parallels. The metabolism is similar. To prokaryote counterparts. Right? They're semi-autonomous as well. Secondary endosymbiosis is also very important. The more greater number of organelles, you increase the number of genomes. So that's important. Semi-autonomous. Own DNA. They have their own DNA. And they do their own stuff. Right? They have their own ribosomes too. They're pretty separate, but it's a pretty good theory, I reckon. Works. Yeah. Okay, cool. So we talked about that. We talked about how it evolved. Now let's talk about different types of eukaryote, plant and animal. Okay. So we're just going to do this very simply. Plant versus animals. And cells. Okay, so plants have big vacuole, very important, big vacuole. Animals have small vacuole. That's an important difference. There's a thing, it can change turgidity and everything. There's a lot of different things about it. But they've got different functions and structures, and another notable one is plants have a cell wall, cellulose. Animal cells have no cell wall. What else can we sort of talk about? Plant cells do have chloroplast. Okay, so just think about this while I quickly search something up to make sure I'm not wrong about something. So, yeah, that secondary membrane in a prokaryote, I mean, when we have primary endosymbiosis, the membrane kind of forms because you have the prokaryote itself, which has a membrane, and then as it gets engulfed into a eukaryote, there's like a new membrane formed, right, around that, and that's your second membrane. Right, and it doesn't dissolve per se, like there's like a thing around it as well as like a vacuole or something, right, because it wants to kill the bacteria, but it's not necessarily like, ¿cómo se dice? Like they're like things, and the cell wants to kill that thing, but it doesn't, and eventually gets integrated in. But yeah, with a secondary endosymbiosis as well, because a cell is being taken, like a eukaryote is being swallowed by another eukaryote, the nucleus is going to be like destroyed of that other one, so like dissolved out. And since, like, think about the membranes here, I'll just write this down. Organelle, two membranes. Makes sense to me, and I'm the one learning it, so that's important. So you have the two eukaryote with organelle. Right, so 2 of organelle plus 1 of old cell plus 1 of new cell. DNA transfers over. So sometimes this new one is destroyed. Not up here though. Up here stays. Right? So you can have a... So you get a end result is a organelle with 3/4 membranes. Sometimes. I mean, and just remember the membrane. The old cell shrinks. Right? Hope that makes a bit more sense when I wrote it like this. This cryptomonads, it's like halfway point. Since the like secondary endosymbiosis as well, because it's like a eukaryote that does the engulfing. Anyway, that's fairly... Apologies for the rambling. The first bits of it were good, but we did lose a bit of focus towards the end. The evidence here is pretty nice though. Could agree. I'm one of those. Okay, so we covered these 3 things. Just This one's a pretty small topic. But yeah, hope you enjoyed. You know, I really think people should just put me on for, like, sleeping. If you like the sound of my voice, which we're getting there. Anyway, thanks very much. Have a good one.